Hello, I'm Colin Kay, Associate Professor of Translational Nutrition at the Plants for Human Health Institute.
And hi, I'm Marianne Lila, and I'm the Director of the Plants for Human Health Institute. And Colin, today I'd like you to just describe a little bit for us your BAM study, your blueberry absorption and metabolism study. Now, most times when you do a human feeding trial with a functional food, it's to look at a disease biomarker or a therapeutic benefit, but yours is different. Can you tell us about that?
Yeah, so we were interested in comparing how you absorb and process things from fruits. And is there any difference between a fresh fruit and a processed fruit? So we did a type of study called a kinetic study, where our endpoints are just how things are processed in the body as opposed to what tissues they act on.
Okay. Well, how would you design that kind of a study? How was that done?
In this case, we were looking at blueberries as a phytochemical-rich food that's very well characterized. As you know, we've done a lot of characterization here at this institute. So we wanted to compare two different varieties, essentially two different accessions. So they were distinct in their phenotypes or their phytochemical compositions, but they were both high-bush blueberries. And we also wanted to compare that to a processed product made from the same berry. So this was a minimally processed protein bar that had one of those varieties of berries in it. So to make that level of comparison, we had to do a four-way crossover design. So we had a controlled diet. Then people were randomized for either control, each of the blueberries, or the blueberry bar, basically. They were on a controlled diet as far as everything else they eat. They ate them at breakfast. And then we took blood and urine samples over 48 hours to trace the movement of the metabolites after consumed.
Okay, so each of your clinical participants would consume each of those interventions at some interval of time.
Exactly. It was a controlled crossover study. So that it took a long time. They were in for multiple weeks, basically. There were washouts in between, but we managed to get 17 participants through the trial.
Excellent. And what kind of outcomes were you looking for?
So we're looking for a few outcomes. One is pharmacokinetic parameters, so that can be anything from things like Cmax and Tmax and half-life and area under the curve, but we're also interested in total excretion or total recovery because one of our hypotheses is that there's no difference in the total bioavailability of each of the berry sources or the processed product, but there's most likely differences in kinetics as a result of the different matrices that they contain. So we had to capture total excretion and a number of other kinetic parameters to test that.
Okay. Your data is not analyzed at this point, but were there any surprises for you?
We just finished the last participant last Wednesday or last week.
Yay.
So we're now in the stage of finalizing our methodology to run. We're looking for over 180 analytes, so we have to make sure that the final method is 100% before we start on the analysis. Because it's a kinetic study and there's so many time points, we have thousands of samples to analyze, so it will take a while. I guess the one thing that is interesting and exciting is we had a clinical team that persevered through COVID. We had significant shutdowns. We could only process at certain points one person a day, and on a 4-phase crossover trial, that's extremely difficult. So it's taken considerably longer than planned, but a lot of clinical studies have been canceled, and we're really proud of the fact we were able to complete this. And it was truly just from having a really dedicated clinical staff.
That's excellent. Well, this was a preliminary study, but what comes next? What would you think?
Yeah, so we chose blueberry in this study just because it's very well characterized and we have a lot of experience working with it, but really it's part of a larger initiative looking at fruits and vegetables. And the concept is that if you have the same fruit grown in two different environments or two different sides of the country, or you take that fruit or vegetable and you process it, are you going to see similar things in the sense that is your blood and your tissues in your body going to see the similar nutrient profiles? So we need to replicate this with multiple food sources. And the ultimate goal was always to establish, can we get people to consume more fruits and vegetables? And if they can't in a raw form, can we deliver a processed form that has the exact same phytochemical profile that's being delivered to the people? And we think that we can, and the goal is just to get people to eat more fruits and vegetables any way that they can. So this is just a proof of concept that we can do this on a highly characterized fruit, and now we need to move on to other fruits and vegetables.
So the take-home message for the consumer is that it's not so important what's in the fruit or vegetable, it's how much it gets metabolized and kinetics through the body.
Yeah, and there's a bit of a misconception that fresh is always better. It's more about how your body releases it from the matrix, how you absorb it, how you metabolize it. So in a lot of cases, processed foods can deliver more value than non-processed foods. There's a lot of things that aren't bioavailable and they need processing to become bioavailable. So it's just really about establishing equivalence of healthy things in our diet, and can we just get people to eat more of them?